Okay, my name is Phil Rosello, and this is exam 4 written answers. Just going to go ahead and start with number 1 here, and this one is asking the overall phenotype of the cell under certain conditions. So for A, I got constitutive, since the I- does not produce any repressor, and the P site is active, which is the polymerase, and the O site is the binding site for the repressor. That one is active as well, but since there's no repressor produced, this one's going to go ahead and make your product all the time. So that would fall under constitutive. For B, you have this IS, which is a mutant repressor, which means that it's not activatable by lactose. So even though the P site and the O binding site, or I should say even though the P site's working, the O binding with that mutant repressor is not going to ever allow the lactose to activate it. So that's going to be non-inducible. And then for C, we have repressor being made, and the repressor cannot bind to the O since it's mutated here. And if you notice here, the P is not active either, so the polymerase is not going to ever produce anything, even if nothing can bind here. So that one I would consider non-inducible as well. For D and E, we have the F plasmid and the chromosome involved, so we need to take into account how they affect each other, if they do at all. So for D, we have a mutant repressor made, which is dominant to the regular repressor, so that one's going to bind basically in place of the regular one. So for this F plasmid, we have polymerase active, and we have a binding site active, so it's going to grab that. It's going to grab that repressor, the mutant repressor, and is never going to be able to be inducible. But this chromosome part, if you notice here, the binding site for the repressor is not active essentially. So even though repressor is being made and it's mutant, it's for both of them, it's not going to bind at all, and the P site is active for this. So this one's going to produce constantly. So that would fall under the constitutive phenotype. And then for E, we have to look at both parts again with the F plasmid and the chromosome. So we have our reducer being made, the polymerase is active, and the binding site is also active. And then for the chromosome, we have no repressor being made, in an active P site and an active binding site for the repressor. So even though this chromosome is not producing the repressor, the F plasmid is. So that's basically going to go out into the cell, and the chromosome will grab that up and bind it on the repressor. So we can almost act as if this chromosome is totally normal, just as it was looking at this. Well, I guess it's not quite the same, but the idea applies that if they're all active for the repressor, polymerase, the O binding site, and the Z's, Y, and A are all plus here, then it's going to act as just regular, which is an inducible phenotype. So that's what I got for E. Then on number 2, you wish to use DNA sequencing using the Sanger method to determine the nucleotide sequence of the molecule shown below. And it has all these different things. Basically, the idea is the primer is going to anneal in the opposite direction. It's going to anneal in the 5' to 3', and we're just going to have our newly synthesized strand with the matching sequences for the GC and the T and the A, and so on and so forth as we go through here. So this one happens to have 16 nucleotides attached to the primer. So I just listed those in the 5' to 3' from 1 to 16 here, just as listed above. And when we put this into a gel that's loaded with the DD, GTP, ATP, TTP, and CTP, essentially each of those bands is going to light up whenever that particular sequence appears from 1 to 16. So if we look at this first one here, we have T, so that'll match up with the T with the DD-TTP, and then the A with the A with the DD-ATP, and then the G with the DD-GTP, so on and so forth as we move down. So this is about what it should look like when you have this reaction mixture, and this is what you should see on the bands of the gel. For part 3, we have 2 mRNA codons that have sequences indicated below in parts A and B, and then consider the wobble rules and give all possible tRNA anticodons that combine to the following codons, and be sure to indicate the polarity of each of the anticodons. So the anticodons are going to line up 3' to 5' every time here, and with the wobble, essentially you have the first 2 placements that are their matching anticodons. So for G it would be C, and for C it would be G, and then this third spot it has the wobble rule, right? So it can also line up with not only just G, but some of the other anticodons as well. So in the case of C, using an mRNA, I believe this one binds with G, A, U, and I, and so just keeping that same sequence, the CGG, CGA, CGU, CGI. So same first 2 codons there, and then the third one can be changed. And then for B, we have 5' AAG 3'. So what we're going to be looking at here is our 3' to 5' again, and then your first 2 here are standard. So A's go with U's and RNA, and so we're going to see UU, and then with G is this last one, so G and C go together, and then G and A and G and U should also go together here. Looking at 2 tRNA anticodons, so this is a little bit different. So instead of mRNA codons, we have 2 tRNA codons, anticodons that have the sequence indicated below in these parts. Consider the wobble rules and give all possible mRNA codons that combine to the following anticodons. So it's kind of opposite of here. So the way they act is a little bit different in that last wobble spot. So for C, we have 5' UAU 3'. So the anticodon is going to be the opposite. So we have 3' and then our letters to 5'. So for this first one, UA is matched with AU. So if we can see for each of these, we have A and U, and then our U matches with A, and also matches with U, G, and C. And then for D, we have 5' AUG 3'. So our anticodons should be 3' to 5', and then A lines with U, and U lines with A. So we have UA for each of these, and then G and C, and G and U for this. Looking at number 4, we have the average mass of the 20 common amino acids is about 125 daltons each. Suppose you have an E. coli protein called your favorite protein, YFP, is 75 kilodaltons. What is the length in nucleotides of the portion of the E. coli mRNA that is translated to make YFP? So if each of these amino acids weighs about 125 daltons each, if we have 75 kilodaltons, which is thousands, so 75,000 daltons divided by 125 daltons, we should get 600 amino acids, and the length of nucleotides of the portion of the E. coli mRNA, so the mRNA needs 3 codons to make 1 amino acid, so it's going to be a 3 to 1, so we're going to do 600 times 3, so we should get 1800 nucleotides in length. The E. coli mRNA that makes YFP contains 5' UTR and 3' UTR. What is the function of the 5' UTR? This is the untranslated region, and the 3' untranslated region. So the 5' UTR is the Shine-Dalgarno sequence, and it is the ribosome building site in translation. And for the 3' UTR, this is after the stop codon, and this actually terminates transcription. For C, we have how many ATP molecules are required to translate 100 copies of YFP, and we know that there is 1 ATP molecule required for every copy of YFP made, so essentially we just take our 600 amino acids, or I should say there's 1 ATP per amino acid required, so we have 600 ATP required for our 600 amino acids, and then we have 100 copies of that, so we're just going to take 600 ATP times 100 copies, and that will give us 60,000 ATP required. And for D, we have how many GTP molecules are required to translate 50 copies of YFP, and the formula for GTP is 2N, which represents amino acids, plus 1. So in this case, it's 600. So you have 2 times 600 plus 1 times 50. So we have 1201 GTP required for 1 copy, and then 50 copies, you just multiply that by 50, and that would give us 60,050 GTP molecules required to translate 50 copies of YFP. And that's what I got for my exam 4 written question answers.